My name is Sumit Goswami, and I actually work for Pfizer at Formulation R&D as a senior scientist, located at Chesterfield, Missouri. Well, like I said, I work for Pfizer, pharmaceutical company. It's pretty well known, but I want to emphasize that we, the scientists at Pfizer, we try to make sure that we create new medicines that have strong potential of changing patients' life, and safety and compliance are priority. At the same time, we try to make sure that quality product is catered to diverse patient population. And as you know, quality comes at the cost of cutting-edge research and strong innovative core. And at Pfizer, the environment is nourished such that innovation is strongly encouraged. So being in line with that mission at Formulation R&D at Pfizer, we are heavily focused at constantly improving our understanding and capability of tackling some of the major challenges the industry is currently facing. Achieving low viscosity at high concentration situation that improves manufacturability and injectability is one such challenge that we are currently dealing with. So my talk in this conference actually is aiming to provide an overview of our understanding of how solution conditions and intermolecular interactions work together to influence viscosity situation. So it's an example of wonderful collaborative work between different departments and colleagues within our department. And that's again in line with Pfizer's mission. So as I mentioned earlier, we are trying to understand the viscosity behavior at high concentration, and we're trying to understand it from a mechanistic standpoint. So, protein-protein interactions plays a major role in controlling viscosity, and protein-protein interaction is largely mediated by electrostatic interactions. Now, there are different kinds of electrostatic interaction: charge-charge, dipole-dipole. So in this talk, we have tried to provide an overview of different types of electrostatic interactions, specifically focusing on charge-charge interaction. And charge-charge interaction, being repulsive in nature, has the tremendous potential of being used as a tool for minimizing viscosity at high concentration situation. So measurement of charge is important. That's one of the messages that I'm trying to give out to the audience. And we will also remember measurement of charge is important at the solution condition of relevance. But the question that we're also asking is charge measurement enough to understand the viscosity behavior, or is there other electrostatic interaction components that are equally relevant in terms of understanding viscosity behavior? So we have made an effort to understand how solution conditions and different electrostatic components interplay with each other to control viscosity behavior. There is a lot more to understand and achieve, but I think it's a good start. First of all, we're trying to introduce a relatively newer technique called membrane-confined electrophoresis. It was originally developed by Dr. Tom Lowy and currently commercially available through Spin Analytical. This has not been used very much before. So what we have done charge measurement using that technique. As the name implies, it's an electrophoresis-based technique. We don't have the scope of really do a comparative assessment of this technique versus the others that are available for charge measurement. But I must say that it does provide some advantages over others, and that's why we have used it for our talk. And the other aspect that I want to mention, once again, we're trying to understand viscosity from mechanistic standpoint. So we have provided specific examples of model proteins which have distinctive viscosity behavior, and we have tried to explain their viscosity behavior with solution condition and electrostatics. So not only we have provided new data, we have also tried to come up with models to explain the data. So as a formulation scientist, things that will keep me busy or engaged over the next few years, first of all, a major focus will be to understand viscosity from its origin. We understand viscosity in bits and pieces. So the key would be to extend our knowledge further to the extent that we can, that allows us to connect the dots between different contributing factors. That's number one. Number two, I would like to have a toolbox for early assessment of candidate at pre-formulation stage. And there are different parameters that are currently being evaluated for early assessment of candidates, including those that are dilute solution properties. So the key would be to assess the potential of each of those candidates and link them to early assessment. And the other one, maybe a bit ambitious, is to perform analytical assessment on directly at high concentration, which I think would be of utmost importance, and I think that's a current gap that the industry is facing right now. So we have to important it's a knowledge share, because at biopharmaceutical industry we are doing a lot of different things, but it is important that we come up and we share those things with other folks. So not only it saves time, and it also allows us to focus on what is important and what other people are doing, and it changes our perspective, changes our way of thinking towards solving a problem. So I think these kind of conference, not only BPI, any conference like this where you have industry and academia are taking part together, is very important for continuous improvement.